Our three major classes of blood vessels are arteries, capillaries, and veins. Okay, and we're going to look at a few different types. One thing to note is you should always remember this. Capillaries is where the magic happens. Okay, so capillaries are the site of gas exchange, and we'll take a look at what is happening there. You should remember from chapter 22 that arteries carry blood away from the heart, veins are carrying blood back to the heart. So if you think about that, when blood is leaving the heart, it is leaving at a very high pressure. Okay, when it's coming back from the heart, it's gotten really, really far away from that pump, and then it's kind of slowly carrying the blood back to the heart. Okay, so that means that we have to remember our general principle that form meets function. So we're going to see some differences and similarities between these different types of blood vessels. So first we're going to take a look at the walls that comprise these blood vessels, because basically what blood vessels are tubes, right? They are the highways that carry blood through the body. Okay, and if we start at the deepest layer, we have what's called the tunica intima. It is the layer comprised of this stuff right here, which is the tissue that lines the lumen of that blood vessel. What type of tissue would you call that? Epithelial tissue, right? It's called the endothelium, and it is continuous with the endothelium that we find lining the chambers of the heart. Okay, so we have this endothelial layer associated with what's called the subendothelial layer, and it's comprised of connective tissue, areolar connective tissue. Remember what we learned way back in chapter 4. We build an organ similarly in the same way. So if we're making a barrier or a lining, we use epithelial tissue. Here it's called endothelium, and then we want to attach it to the tissue beyond it using connective tissue. Here we're using areolar connective tissue. And then we have the next layer is called the tunica media. And you can see here a major difference between arteries and veins is the size of that tunica media. So before we move on, I did forget to mention you also have this internal elastic lamina that you find in arteries and not in veins. So arteries are generally a lot more elastic than veins. Okay, and if you think about it, they are receiving, especially the big arteries, the elastic arteries. There are arteries called elastic arteries, like the aorta. It's going to be receiving these high pressure pulsations of blood every time the ventricles contract, right? So they have to be able to stretch and recoil to receive those high pressure pulsations of blood. Okay, so you find this internal elastic lamina, and it's comprised mainly of elastic fibers. All right, then you have the tunica media in both arteries and veins, and you see that in arteries, it's a lot thicker. That tunica media is comprised mostly of smooth muscle, right? So you have smooth muscle followed by, just superficial to that, in arteries, you have an external elastic lamina. So in arteries, as opposed to veins, you have two layers of this lamina with lots and lots of elastic fiber that allows the artery to stretch and go back, stretch and recoil in order to receive those pulsations of blood. Whereas veins do it a little bit differently, right? And we'll talk about veins. You can see here, some veins have valves to prevent pooling of blood and to push blood back to the heart, okay? That tunica media is also under the control of the sympathetic nervous system, right? So when we want to do our fight or flight response, when we talk about shunting blood from certain areas or changing blood flow to other areas, what we do is we constrict this smooth muscle right here. And if you were going to constrict that smooth muscle, what do you think would happen to the lumen of this blood vessel? It would constrict, right? That's called vasoconstriction. So you contract the muscle and you constrict the lumen, vasoconstriction. And when that muscle relaxes, you have vasodilation. All right. And then next we have the tunica externa, or the superficial most layer of these vessels, and it's going to be comprised of some kind of epithelium as well as connective tissue in order to anchor it to all of the surrounding tissues, right? You also have the vasa vasorum. So that literally means vessels of the vessels. Just like in the heart, we talked about coronary circulation. Within blood vessels, they are also organs and tissues that need their own blood supply. All right, so that's what the vasa vasorum does, and you find that in the tunica externa. Okay, and then leading from arteries, as we travel from the heart, arteries get smaller and smaller and smaller until you get to capillary beds, and we'll take a closer look at this. And then they travel back at the end, the distal end of that capillary bed, to veins and back to the heart, right? And capillary beds are microscopic structures. They're really, really teeny tiny, and they only allow for that single file of erythrocytes called the ROLU that we saw in chapter 21. All right, and they're just comprised of, so this is one of the few places in the body where you just see the endothelial layer followed by a basement membrane. You don't have any of that internal connective tissue, all right, because this is the site of gas exchange. You want things to be able to diffuse quickly in and out. Okay, as we go through this chapter, you will pick up more and more detail, and my brain will function better as we get through the material. But one thing to note is that blood is going to move really slowly once it gets to these really, really highly branched capillary beds, right? You want the blood to move slow enough because gas exchange happens via diffusion. Right? And you want it in order to equilibrate with, you want the blood inside the lumen to equilibrate with the fluid outside of the lumen, where you're going to have oxygen diffusion out of the capillary beds and carbon dioxide diffusion back into the capillary beds. So hopefully by the time you get to the end of this capillary bed, you have completed doing your gas exchange, right? So really tiny, tiny, tiny lumens, and they're highly branched so that you can get to all of the tissues in that area as well as slow the movement of blood through that area. Okay, if you look at a microscopic comparison, this is like one of the really cool things that you can observe in anatomy, where it kind of looks similar to the stylized cartoon. If we look at a vein, if I was to show you this on an exam, you should immediately be able to recognize that one of these is a vein and one of these is an artery. Because if you look in the vein has a very large lumen, right? And the blood pressure within veins is a lot lower. Okay, when we talk about blood pressure and movement of blood through arteries and veins and all these vessels, you can think about how water moves through an irrigation system, right? If you turn on the faucet and you start with a really big hose with a very high pressure, and then you eventually branch out, it's going to move slower and slower as you branch out. Okay, so fluid dynamics is a really good way to remember how blood moves through blood vessels, right? So you see here, the walls of the vein are a lot thinner, whereas the walls of the artery are much, much thicker. We have a lot more smooth muscle within the artery, and we have a lot more elastic fibers within the walls of the artery. All right, so we have different types of arteries and veins, and we classify them based on size and the components of their walls that make up the walls, right? So in artery land, okay, our biggest artery is called an elastic artery. And from its name, we know that it is very elastic. It has a lot of elastic fibers, okay? And it has a very, very thick tunica media, okay? One type of elastic artery would be the ascending aorta. They're also called conducting arteries because they are conducting oxygen-rich blood to the rest of the body. So they are the ones, like the aorta, like the pulmonary arteries, they are receiving these high-pressure pulsations from the ventricles and sending them out to the rest of the body. Okay, so we have lots and lots and lots of elastic fibers, and that's your aorta, your pulmonary arteries, your brachiocephalic, your subclavian. We will learn all about those. Those are elastic arteries. As we get farther away from the heart, we go down the descending aorta. So some of you were confused in the previous chapter, ascending versus descending aorta. It's continuous, right? I'm going to change my color to yellow. Okay, this is the ascending aorta. Then we get to the aortic arch. It's like the bottom of an upside down U. And then it descends through the thoracic and abdominal cavities, right? It's this one continuous structure. And if you were in lab, you would be able to see this really, really cool, very large blood vessel traveling through the length of the anterior compartment of the body. As it gets farther away from the heart, okay, we call them muscular arteries, and it is because they have lots of muscle. Those are our distributing arteries. So those are the arteries that are named, like the brachial artery, the anterior tibial artery. Those are the ones that are carrying blood supply to eventually to arterioles and capillary beds. So those are smaller, right? They're on the order of millimeters, right? The elastic arteries have a diameter of a few centimeters, like 1 centimeter, 1 to 3 centimeters. Right, as we get into the muscular artery, we're like less than 3 centimeters to the order of millimeters. And then we get into arterioles. So when we get to arterioles, you see a structural difference where you don't have as much smooth muscle, and you also you have you could have an endothelial layer with just one layer of smooth muscle. Generally, arterioles have very, very thin layers of smooth muscle. Okay, the arterioles are going to lead directly to the capillary beds. Okay, that tells us something. The arterioles are the site of control of blood supply to the capillary beds, and that's really, really important for distributing blood to different areas in the body, right? We don't have enough volume of blood to have all of our capillary beds being supplied at any particular time. We're always shunting blood to different areas depending on demand, right? Like if you are running away from a saber-toothed tiger, you're going to shunt blood away from your digestive organs because it's not time to digest that yummy, delicious egg, cheese, and hash brown sandwich that you had in the morning. It's time to shunt blood to the skeletal muscles in your legs and in your arms, and the smooth muscle that contracts and relaxes to allow you to inhale, right? So we're constantly controlling movement of blood within the body, and that's how we do it, by changing the supply to different capillary beds. Okay, and we'll take a closer look at that. Okay, when you go through the capillary bed, and as you get back to going back towards the heart, you're going through veins. Okay, no longer have, they're not as elastic. They don't have the layers of elastic fibers that arteries do, and they have much wider lumens. So the blood is kind of moseying on back to the heart. The receiving vein from capillary beds is called a venule or small vein. And then as you get larger, you go into small to medium-sized veins, and those are most of the veins that we name. And then you get into large veins. The large veins are the two major ones that dump into the right atrium are your inferior and superior vena cava. Okay. Escape. Go next. There are your words. There are your words. There are your words. So you should be able to compare and contrast arteries and veins. Okay, what is their function? Here are histology slides of different types of arteries, right? You can see that in the elastic artery, you have this very, very thick tunica media, right? It gets a little bit thinner in the muscular arteries, and then in arterioles, we have very, very few layers of smooth muscles. It's like 1 to 6. And this right here, this little purple stuff right here, that is your smooth muscle. Okay? Okay, supply of blood to a capillary bed goes from an arteriole and into this structure called a meta-arteriole. It's a smaller arteriole, and as you can see, they're surrounded, these arterioles and meta-arterioles are surrounded with smooth muscle that can constrict and change the amount of flow that you have within the capillary bed, right? So first thing we should notice is that there's a lot of branching, right? We want to be able to reach all of the tissues, and just in case we can't, there's something called an anastomosis, and there's a really, really important one that we're going to talk about in the brain. We talked about anastomoses in the context of coronary arteries because they don't have enough anastomoses, so there are overlaps between blood supplying vessels that allow for oxygen-rich blood to reach a tissue in case you have blockage somewhere. All right, that's why coronary arteries are called functional end arteries. They do have anastomoses, but not enough that if you had blockage, you would not die. You would die, right? So as the blood travels from the proximal end of the capillary to the distal end through this thoroughfare channel, it by diffusion is going to drop off oxygen and pick up carbon dioxide. And capillary beds have something like 10 to 100 capillaries within a particular capillary bed. And the way you can actually shunt blood from this capillary bed is through these precapillary sphincters. Okay, those are circular smooth muscle that can constrict and push blood more quickly through this thoroughfare channel, so it's shunting blood from that capillary bed, right? And that's what would happen. So if your sphincters are contracted, blood is going to bypass that capillary. Okay, if we look at different types of capillaries, there are different types of capillaries, sorry, and they're classified based on the structure of their endothelium. So we have what are called continuous capillaries, and those are the most common. You see them in muscles and the blood-brain barrier, and they have this continuous layer of endothelium, and it's directly attached to the basement membrane, right? So you don't have any gaps. And if you didn't want anything to pass through this layer of endothelium unregulated, what types of cell junctions would you put there? if you said tight junctions, you are correct, right? So they're attached to each other. They're called continuous capillaries because they don't have any gaps in the endothelium. There's another type of capillary called a fenestrated capillary. And fenestrated means a window. A fenestration is a window. So they have holes or windows within the endothelium, allows them to absorb or it allows passage of more things. So somewhere where you would want to allow slightly more unrestricted passage of molecules would be somewhere like in the small intestine or the kidney, right? So they're not completely continuous. So you have your layer of endothelium associated with the basement membrane. And the last type of capillary that we see is called a sinusoid. And just looking at this structure, this cartoon, even the basement membrane is discontinuous, right? So you have your layer of endothelium, right? And that's not continuous with each other. You have large gaps in it. And then you have even the basement membrane is not continuous. Where in the body would you want something like this? Where you would want to kind of have a very free flowing of substances between the lumen of the blood supply to the outside, not the brain, right? You would want it in your water sanitation or your sanitation station, right? That would be in the liver. So the blood supply that goes from the hepatic portal system, from the digestive system to the liver is going to be carrying lots of pathogens and bacteria and things like that. So you want to be able to have access. You want to be able to allow your immune system to attack foreign pathogens. So you do find sinusoidal capillaries in the liver. Gosh. Okay, and last but not least, last part of this lesson is the structure of veins. So venules, we have venules receiving blood on the other end of a capillary bed, and then as they get larger, I think when you get larger than 100 microns is when we call it a vein. And veins are the vessels that drain blood to the heart. Okay, so deoxygenated blood is moseying its way back to the which chamber? Right atrium. Good. Okay, so one of the ways that we actually control, so at any given time, the veins are going to carry around 60% of the volume of blood in the body because they don't have these thick and elastic and muscular walls. They're very thin walls, large lumens, low blood pressure. So we call them the blood reservoirs. But they do have valves. And there are two types of pumps that are really cool that help to push venous blood back to the heart. One of them, so you have these flaps that are kind of like one-way valves. Sound familiar? That allow blood to travel past them, but not, it allows for unidirectional blood flow. So once the blood passes this valve, the pressure on the other side of the valve is going to prevent blood backflow and keep blood moving to the heart. Okay, there are two types of pumps in the body that help pump blood back to the heart. One of them is called the muscular pump. So these veins, the deep veins, are going to travel in between skeletal muscles. And as we contract our skeletal muscles, it's going to push on the vein walls and push blood and pump it back to the heart. So those are skeletal muscle pumps. And the other one is a really, really cool one. It's called the respiratory pump. And that is how we push blood through the abdominal, pelvic, and thoracic cavity back to the heart. Okay, so think about this. When we inhale, we contract the diaphragm and we flatten the diaphragm. We increase our thoracic volume, the volume in the thoracic cavity by inhaling, right? And we decrease the volume in the abdominal pelvic cavity. By decreasing the volume in the abdominal pelvic cavity, you're increasing the pressure and causing compression of blood back in the veins through the inferior vena cava back to the heart. Okay, that's when you inhale. Then you exhale, you decrease thoracic volume and increase thoracic pressure. You release the compression of these guys in the abdominal pelvic cavity, and you increase the pressure in the thoracic cavity, putting pressure and causing compression on the inferior and superior vena cava and all of the veins leading back to the heart. All right, so you inhale, push blood up from the abdominal pelvic cavity into the thoracic cavity. Then you exhale and push blood from the thoracic cavity back to the heart. All right, so really cool way that we actually move blood through veins. So that is it for this lesson. Anatomy, this corresponds to chapter 23, part 1, the anatomy of blood vessels, comparing and contrasting different types of blood vessels. And then this last part was factors that influence venous return. Okay, so even though veins have different structures than arteries, they still have a anatomy that allows them to do their physiology, and their function is to drain blood back to the heart. Okay.